Hello everyone, and welcome to today's webinar, Immunotoxicology Assay Toolbox, Maximize Standardization. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. Urmi Roy, Global Product Manager for Flow Cytometry Reagents at Miltenyi Biotec. Urmi, you may now begin your presentation.
Hello everyone. Thank you for joining us in today's webinar. Today I will discuss with you about immunotoxicology, one of the very important segments within drug development. In today's presentation, as for introduction, I will discuss about immunotoxicity, mainly different assay consideration, common assays, and extensive validation of those assays that are proposed by regulatory authorities. I will then focus on experimental tools, specifically flow cytometry for assay analysis, and how we can support assay standardization with our solutions. Finally, I will show you some of exemplary data that are of specific interest for immunotoxicity assessment, mainly immunophenotyping, NK cell activity assay, and cytokine measurement. To start with, it is no surprise to you that our complex immune system encompassing both innate and adaptive immune system maintain the balance between host health and disease. With our increasing knowledge of host immunomodulation, we are now capable of manipulating these to obtain improved therapeutic outcomes. As a consequence, we see increased presence of immunomodulatory therapies in the field. As you all know, significant number of drug candidates fail in the clinical trial due to safety issues. Drug safety assessment, therefore, is a very important battle to ensure successful development of a therapeutic candidate. Interestingly, immune system was not often considered for safety pharmacology studies. With increasing number of immunomodulatory drugs, unwanted immune responses were also more evident. There are different immune-related adverse effects that can be triggered by drug candidates, such as immunostimulation, which results in excessive immune response related to, for example, cytokine release, flu-like reaction, etc. And this can be commonly observed. There are also effects like immunosuppression, which includes infectious complications as well as virus-associated neoplasia, can be observed mainly in immunocompromised patients. Immune-mediated allergic reactions can also be observed. Although rather rare, systematic autoimmune responses can appear as adverse effect from immunomodulatory drugs. With having all such adverse effects in mind, at the very end of 20th century, regulatory requirements for non-clinical immunotoxicity evaluation of pharmaceuticals started to rise. Currently, ICH S8 guidelines provide systematic incorporation as well as the core requirements for immunotoxicity studies for biopharmaceuticals, which is followed by both FDA as well as EMA. This guideline nicely outlines all the detailed consideration one needs to think about when designing immunotoxicity studies. The initial consideration for immunotoxicity studies depend on standard toxicity study results, which include assessment of 4 key observations. The hematology observation includes measuring the total as well as absolute differential leukocyte count. So one needs to check the absolute count of these cells. Then there is also the aspect of clinical chemistry, where the commonly looked parameter is globulin levels as well as the A and G ratios. One also needs to look into the gross pathology changes, such as for lymphoid organs, tissue, organ weight, and as well as for measuring the thymus and spleen in terms of changing the weight of this. Also, optionally, different lymph nodes can be measured. The last parameter is this histology aspect, and the recommendation are there for looking into thymus, spleen, and draining lymph node, and to at least one of the additional lymph node to check the histology properties of these specific organs. Additional immunotoxicity study consideration comes next, and the initial consideration depend on the results obtained from standard toxicity studies. And to conduct additional immunotoxicity studies, several factors needs to be considered, mainly based on the weight of evidence, as I mentioned, from the standard toxicity studies. Other factors include pharmacological properties of drug. For example, of course, if the drug mode of action is immunomodulation, there is higher chance of observing adverse immune effect. So this should be considered. Then the intended patient population, as I mentioned earlier, immunopatient that's suffering for immunocompromised disease will more likely show more adverse effects. So such population also needs to be considered. Structural similarities with other known immunomodulator also is an indication for additional immunotoxicity study consideration. Moreover, disposition of the drug as well as the clinical information can support the fact that additional immunotoxicity studies are needed. Additional immunotoxicity studies typically consist of repeated dose effect, at least 4 weeks. The most common assay that is considered as the first line of assay is known as T-cell dependent antibody response, that is TDAR assay. And this assay can assess the global immune function. Commonly used assay here is anti-KLH antibody response, which is measured by ELISA, typically done in rodent model. Second line of assay include measuring lymphocyte subset via immunophenotyping. Although this assay is not a strict immune function assay, the result offers valuable insight about immune cell subset composition, which could give specific response. If these two assays suggest specific cell subset independent specific cell subset dependent toxicity response, then further assays are recommended to confirm those specific response. Such assay includes, for example, NK cell activity assay, where one checks the cytotoxicity, cytotoxicity feature of the NK cell. Further on, host resistance studies are also performed, where factors like tumor proliferation can be monitored. Further on, some in vitro macrophage as well as neutrophil function assays are also suggested, where one can look into the phagocytosis, oxidative burst, as well as cytolytic activities of these cell subsets. Last but not least, there are also recommendations to measure assays for cell-mediated immunity, specifically delayed type hypersensitivity reaction. All these different types of assays are different test model can be used. For some, there are of course in vitro studies can be performed. In many cases, these are recommended to run this assay in rodent model. Also, non-human primate model are considered. Sometimes, depending on the drug compound, canine models are also used. Now, to ensure the confidence of this assay result, extensive validation is required and as well as recommended in these guidelines. Such as what can affect this kind of variation is inter and intra-assay precision. There is also increasing concern about variability coming from different operator, that indicating technician to technician precision, as well as the accuracy and sensitivity of the assay. One needs to validate extensively. Specificity of the assay is also important. One needs to see the robustness, linearity across different time, and of course, even though the studies are non-clinical at the non-clinical stage and it bridges the connection to clinics, some of the assays are also suggested to done under GLP environment. Variation in assay can result in significant economic impact. A publication in 2015 in PLOS Biology reported that 50% of preclinical research is irreproducible, resulting in a waste of almost $29 billion US. The sources of such variability is also categorized differently. The top culprit of which is biological reagents and reference material, which refers to almost 36% of the irreproducibility reason. Also followed by study design that can also affect irreproducibility, causing almost 27% of as the reason. The third variability comes from data analysis as well as the way it's reported. 25% of the reason of irreproducibility comes from data analysis. And finally, a correct protocol in the lab that is followed results in 10% of irreproducibility. Now, what one could do to avoid all this irreproducibility and utilize the better experimental tool to ensure standardization. As I mentioned before, I will particularly focus on flow cytometry technology for immunotoxicity assay analysis and also our solutions for that. Flow cytometry offers several advantages why it could be used for immunotoxicity assay over other analysis technology. Some of them are listed here. Some of you are already aware that, of course, this tool offers you a single cell resolution. One can perform assays in a higher throughput, also obtaining higher content. You can zoom within the subpopulation and get more in-depth data out of your experiment. Of course, one can analyze the entire population of cells in a higher throughput, and this tool also allows you to multiplex as well as it is compared to some other available technology, it can be also easy. If you remember some of the assays that were shown previously, as an analysis of some of those assays, flow cytometry can obviously be used. You can, as a first assay, you can think about immunophenotyping. However, not only immunophenotyping, one can also perform flow cytometry analysis to measure cytotoxicity, also proliferation, cell proliferation, and then to measure the cell counting at subcellular level. Flow cytometry is also really excellent tool. Also, this advanced flow cytometer allows you to monitor phagocytosis and oxidative burst. So all in all these different aspects can be measured by flow cytometry. Now, of course, this flow cytometry analysis also comes with a lot of variability, some of which I already mentioned earlier, that is associated with the assay variation. Here, of course, one of the main variation is coming from the donor. So the sample that you are measuring via this analysis tool coming obviously from different donor, and this, of course, is one of the source of variability. Then what type of protocol and panel design one is considering can vary from lab to lab, person to person. especially if you remember, the study also showed reagent is one of the important factor which contributes to assay variation, which comes along with the lot-to-lot consistency and specificity variation. Flow cytometer instrumentation also can cause variability, especially due to operator handling the instrumentation and doing different type of pipetting and so on. And finally, also, of course, analysis and reporting can vary from person to person. Of course, donor variability is a very difficult aspect to tackle, and this is something one needs to carefully consider when setting up the assay, and something of which we are not able to support, but at Miltenyi Biotec we offer a lot of solutions for flow cytometry which can support the other aspect of the variability for your immunotoxicity assay. To start with, one of the important reasons for immunotoxicity assays are antibodies, of course, mainly for your immunophenotyping. Miltenyi Biotec, through its pioneering technology, offers recombinant antibodies since 2012, which offer the best quality immunophenotyping results. Our antibody portfolio particularly targets the most commonly faced challenges in the field, such as the bad antibody, low antibody quality due to its lot-to-lot inconsistency, unspecificity, also less sensitivity, and also you might use antibody which are not validated for its intended application. There are also reported challenges faced by customer in the field that comes from unspecific background signal mediated by Fc receptor, and some of the complexity in terms of experimental planning and our outdated hybridoma technology. Our reaffinity antibodies, with its unique feature of one isotype backbone as well as mutated Fc region, offers this additional benefit and can help you to tackle all these challenges. What do I mean? I would like to show you here one example data specifically for your NK cell analysis, as NK cell also is an important component and can be used for immunotoxicity studies. So here I show you two different data sets where our recombinant antibody was shown to perform better. So in the left-hand picture, you can see the CD56 that is conjugated to different fluorochrome. And the left three plots are coming from our reaffinity antibody conjugated to our traditional FITC dye, as well as our brighter and better VioBright family dye. And you can clearly appreciate a very nice clear response of this CD56 antibody conjugated to our bio dyes. The right-hand plot shows you a clear example of unspecific response. Here you can see CD56 and KIR2 DL1 antibody staining in a sample, and what you are expecting is that all the cells of NK cells will be double positive, and on the right hand side you can see that there is an unspecific response which is CD56 negative, and this potential is coming from unspecific binding with some Fc receptor. So what you can see here, that in combination with our biodyes, of course, the antibodies are giving you enhanced brightness, as well as this mutated Fc receptor of our reaffinity antibody allows significant reduction of unspecific background signal. Finally, about our antibodies, I want to highlight that we offer more than 16,000 fluorochrome conjugated products that are cross-reactive to different species, not only human, but also different rodent species, canine as well as non-human primate. Also the portfolio covers all the major markers to identify different cell subset, immune checkpoint, marker for cell signaling, cell death, proliferation, and so on. And we also provide specific list detailing our antibody cross-reactivity specific to different non-human primate species, which is where varieties of our antibodies can be easily selected for non-human primate toxicity studies. Miltenyi Biotec also offers ranges of flow cytometer known as MaxQuant analyzer family that offers ultimate automation and focuses on operator-induced variability. Our flow cytometer ensures automation through its simple use by automated startup, shutdown, and some of other routine calibration and QC measurement. Also, it reduces operator variability through sample labeling and processing in an automated manner, and we also support with automated data acquisition and analysis. Within our MaxQuant family, we have a special MaxQuant, which is known as MaxQuant X. This is also can be used for high throughput assay and can give you result as fast as analyzing 96 well plate in 15 minutes. All our flow cytometer offers very precise data, so all this automation does not hamper the quality of your data, and it ensures as minimal carryover in the field that is available, as well as support the absolute cell counting, which I will show you a little with one slide later, and the other feature of consistent result through unibody optical bench and multi-instrument alignment I will also highlight shortly. Since many of our customer in the immunotoxicology field appreciate this absolute cell counting performance of the MaxQuant flow cytometer, I would like to highlight this also here in this webinar today. In this plot that is you can see right now, this plot shows you the cell counting performance of 100 MaxQuant device produced consecutively and how it performed in regards to its precision of cell counting. As you can see, the data shows highly precise and reproducible cell count only with 0.95% of CV. And here is another data set which shows you which was generated to monitor the linearity of the cell counting performance. And from this data set, you can see that our device demonstrates nearly perfect linear counting over a large dynamic range. So this could be of high benefit for immunotoxicity studies where you care for absolute cell counting. I would like to highlight quickly another feature of the cell analyzer that is important for accurate immunotoxicity studies, that is multi-instrument alignment. When you require to have your assays transfer across department and need to ensure performance over time, our instrument ensures this multi-instrument alignment through calibration and QC, as well as transferring the consistent result dependent on user, instrument, and location. And this is done through transferring the exact settings of the instrument from one instrument to other. And I would like to share with you here one of the interesting data set, which was generated by 5 different operators who did not have flow cytometry expertise that much, and they ran the same sample in 5 different MaxQuant indicated here as different numbers. And these devices underwent with this multi-instrument alignment. And as you can see from this overlay plot, that all these instruments generated exact same result of the analysis performance, confirming this accuracy of data over instrument and operator. Regarding assay analysis, I would also like to point out our Ph.D. compliant software, which can be important for immunotoxicity studies, especially under GLP environment. For proper data documentation, our MaxQuant device software is equipped with 21 CFR Part 11 for data acquisition and analysis. You can document user management, audit trail, as well as electronic record very nicely, and can demonstrate this to your regulatory authorities very nice way. The other part of the data analysis support that I would like to highlight is, of course, if you think about flow data, gating is one of the problems that originates from who is analyzing the data. This data set, as you can see in the screen, clearly shows that that with the shift of the positive population, one needs to consider how to set the proper gate, and this will obviously vary from who is analyzing the data. To solve this problem, our Max Quantify software is equipped with a feature that we call is Express Mode, which is an algorithm-based analysis flow cytometry analysis tool, and this is supporting automated data analysis, resulting in simplicity as well as avoiding human error by analyzing the data by different person. So thereby also enhancing the standardization as well as reproducibility. And what is also nice then, of course, it's quick and robust analysis can be performed and can be easily integrated into your SOP. I'll give some exemplary data in the end to show you how this can be helpful for your immunotoxicity studies. So these were all about all the product features and how this can be with some of the quick data, but I of course want to show how these tools can support you with your immunotoxicity assays, and therefore I picked few exemplary data set to show you how together all our flow analysis solution can help you better to design as well as run your immunotoxicity studies. So the first example includes here a basic immunophenotyping. Here, human peripheral blood samples were stained with antibody panel. Here you can see from the list that consists of full recombinant antibodies, and the markers were chosen to discriminate general subsets such as T cell, B cell, NK cell, and more. And using our MaxQuant as well as this available immunophenotyping express mode, this is a clear exemplary data that you can expect by using this automated data analysis, where cells are gated automatically based on their subsets, and this is defined based on the scatter plot as well as the subset definition based on the specific marker expression. You can also expect to see a accompanying data table, which you see on the right hand in the bottom, that elucidate further details on the cell subset percentage as well as the absolute. It offers you the absolute cell count features. So if you use our antibody panel and then use this in combination with the express mode and MaxQuant, you will be able to analyze a large sample subset much more automated manner, and you will be able to get this data set in a more concise and easy way, and can also avoid operator dependent variability. Next, I want to highlight you also that immunophenotyping within immunotoxicity studies, of course, can span over not only general immunophenotyping, but also more subset-specific phenotyping. And one of the problems that comes with the variability, that information source can be variable, and this can complicate the setup of a panel, particularly to decide what type of markers to choose, which antibody fluor combination will work, how to set up gating, etc. And we understand this problem in the field, and to address this problem, we started to generate an antibody panel collection. And this is known as our Miltenyi Biotec tested panel, where currently we have nearly 40 antibody panel covering different cell subsets. These panels are established and tested by our internal R&D experts, as well as kindly provided data by our customers. And here I just wanted to show you what kind of data you can expect, and therefore I showed you an example of a mouse eosinophil and neutrophil phenotyping panel that we have, where you can see the panel composition. So the top right box shows you this antibody panel, which includes what marker was used, which clone was behind this marker, as well as the fluorochrome combination, and what was the purpose to use this marker. And then you can expect to see how to set up a gate to define certain cell subset. And of course, it is coming together with a detailed protocol of staining protocol to so that you can just directly copy this protocol. And we hope that with this panel collection, which we are also continuously growing, this can support the scientific community with their further studies. The next example that I want to highlight is NK cell activity assay. This particular assay was performed in our R&D, and the assay setup was shown as you can see it here in the graphical illustration. First, NK cells were isolated from peripheral blood via two different methods, both using our pioneering magnetic bead-based cell isolation method. One method included untouched isolation of NK cells using CD56 microbead, and isolated NK cells were then cultured with different target cells with different ratio, and cytotoxicity was measured using flow cytometry using propydium iodide labeling. Of course, the other method was isolating the NK cells using a straight form method, and the next assay followed the same principle. Here you can see the data. So the left hand, the flow cytometry plot here shows you the different isolation method resulted in slight difference on isolated subset. The straight from method resulted in enrichment of CD3 positive expressing iNKT cells, which were missing in the untouched method. Cells from different isolation method also resulted in slight difference in cytotoxicity results in both dose-dependent direct killing effect here on this middle plot, down in the middle plot, as well as on the antibody-mediated killing effect. So you can expect also, as I mentioned earlier, you can also use flow cytometry to just directly measure this cytotoxicity at a single cell level. So this is basically highlights this feature in such an NK cell activity measurement assay. For the last example of cytokine measurement, we used our bead-based method to measure multiple secreted cytokines quantitatively. To those who are not familiar with how this method works, here is a quick outline of the principle on the left-hand side outline. So basically, secreted cytokines are captured by fluorescently labeled bead coated with capture antibody, and then you stain it with anti-cytokine antibody, and you can also use a detection antibody labeled with a fluorochrome, and then all these different bead population that are coated with different detection reagent can be separated and therefore multiplexed. And this feature comes in combination with also the express mode that I highlighted before. So you can simply use our MaxPlex reagents and then use the express mode analysis tool together with our MaxQuant. And this allows you to generate a very nice automated assay analysis, which can be, of course, complicated when one needs to do all this gating manually. So the middle plot here shows how this analysis tool helps to generate a standard curve for the cytokine for different cytokine in the assay, and the last plot on the right hand side shows an exemplary data that is generated with the express mode to calculate the cytokine concentration of each analyte of the unknown sample. Now this technology was applied to this for the cytokine measurement in a mixed lymphocyte reaction assay, and this data was kindly provided by our customer, and assay was done in a mouse model. So the assay setup looks like this, where splenocytes, so CD3 positive T cells were isolated from splenocyte, and then they were co-cultured with also splenocyte from a T cell depleted mouse. And then after this co-culture experiment, of course, some interesting compound were added to monitor how it changes the cytokine profile. And this cytokine measurement was done using our MaxPlex kit in a MaxQuant using the express mode. And here is the list of cytokine that were measured together in a multiplex manner. I'm just showing you all the data that customer provided where they found it interesting in terms of different compound. So they basically co-cultured with different concentration of their compound of interest and observed dose-dependent cytokine response, specifically GM-CSF, interferon gamma, and IL-17. And of course, as I mentioned, with this automated tool, you can expect to see such dose-dependent cytokine response in a rather automated manner. With this, I come to my last slide of the presentation. Here, I would like to highlight the summary that, of course, standardization of assay is very important, and specifically also immunotoxicity studies, and optimized tool can be used to better support the assay standardization. And of course, at Miltenyi Biotec, we help you to improve these assays and enhance standardization by offering our recombinant antibody portfolio that is across different species and also highly reproducible in terms of giving you immunophenotyping data. We also showed you how you could use our automated flow cytometer tool to reduce operator-dependent variability and also obtain high-quality data, including accurate absolute cell counting. As I mentioned, also it is if you are performing your immunotoxicity assays under GLP environment, our FDA-compliant software can help you there. And express mode solutions could offer operator-independent assay analysis and enhance thereby the standardization. Finally, we really believe that our tested panel that I showed you earlier, this collection of panel can support you to establish your panel, thereby saving your time required to extensively validate your immunophenotyping panel. If you want to, of course, see more and learn more what solutions we provide, you can go visit our website or can contact us also directly. With this, I would like to thank you all for listening to me, and I hope some of this information was helpful to you. If you have any question, I would be very happy to answer them, and otherwise, of course, you can get back to me later. And thank you, and I wish you everyone a nice day.
Thank you, Urmi, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what are the differences with untouched versus straight from protocol of NK cell isolation?
Thanks for the question. Of course, I had to go quite fast with this step. I think it's a really good question to. So we have, of course, our pioneering technology using magnetic bead to isolate certain cell subset of interest. And in this particular experiment, they have used two different methods. One was, basically targeting to isolate the NK cell by specific marker like CD56, which was straight from. And for the untouched method, there were negative markers used where to not to touch the NK cell as it is. So you basically get out of all the unwanted cell subset. And of course, if you pick a specific marker, CD56, which is also expressed by some other subset, therefore you get to see this CD56 expressing iNKT cell subpopulation within the straight from isolation protocol. And depending on what is your expectation to see in terms of cytotoxicity performance of the cell subset, you can go ahead with any of this method. Should work fine.
Thank you, Urmi.
That was.
Oh, I'm sorry.
No, I just wanted to say I hope that was clear. I mean, otherwise, if there is any question for detailed protocol, you can, of course, write. I mean, I'm not expert in many of some of these essays because it was done by our R&D counterpart, but of course, I would be happy to, provide more detailed information if that is requested.
Thank you, Urmi. Let's see, our next question here. What type of documentation is offered with recombinant antibodies, and what kind of validation is done to check performance?
Thanks for the question. So our recombinant antibodies we offer as a research use, so research use only product, and there we offer this the standard documentation, which is certificate of analysis. And if you purchase our antibody, it's very easy. You can go to the website and download this certificate of analysis. We also offer to customer who's interested to get more requires advanced document and certain type of specific information in regards to concentration of the product and so on. This can also be offered. This can also be handed out to customer upon request. In terms of validation, of course, we have state-of-the-art validation that follows the production as well as the quality control of the product. In specific to a recombinant antibody, we also perform a lot of extended validation to kind of ensure the specificity of the antibody in terms of what target they are recognizing, and via epitope blocking experiment. And we also support extended validation data with some of our antibodies with the knockout cell line, and also we compare one-to-one performance of our antibodies. So there are a lot of validation data available for many of our product line. If you are interested, you can also, of course, find them in the product page.
Thank you, Urmi. We have one more question here. How are the gates set with express mode?
That's a good question. I guess our software specialist would be the best person to explain really nice how this really nice function works in the software. So basically, it works with algorithm, and they analyze more than 300 samples for specific express mode. For example, the one I showed for immunophenotyping, they measure more than 300 sample subset and try to basically with advanced algorithm to try to see where the population should lie within certain marker distribution. And so based on that, the gate is basically defined, and of course to define the subset, the gating is taken from what is typically used in the field. Yeah.
Great. Thank you, Urmi. Do you have any final comments for our audience before we go?
So, of course, as I mentioned, it was a presentation in a nutshell, and if you are interested to learn further, especially the application example that were shown, due to time limitation, of course, I did not go too much into detail. There are also much more application data available that are tested and also validated. Some of them are by our R&D colleague. If you are interested, please reach out. And also, if you're interested for the presentation, the slide deck, I would be happy to send it via email.
Well, thank you again, Dr. Urmi Roy, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you.
Thank you.